Hello everyone. In this lecture, I'll be covering the mTORC signaling pathway and how it relates to lysosomal positioning. I'll wrap up this video by reviewing this fantastic paper by Hong et al. Titled PTDINS3P, which stands for phosphatidylinositol 3-phosphate, controls mTORC1 signaling through lysosomal positioning, published in the Journal of Cell Biology. I hope you enjoy. So mTORC stands for mammalian target of rapamycin complex, and it's a highly conserved protein that was originally discovered to be the enzyme inhibited by the rapamycin molecule, hence its name, mammalian target of rapamycin complex. Now, this rapamycin molecule was first extracted from a bacterium on Easter Island, and it's very fascinating because it was soon discovered to have very potent immunosuppressive and anti-aging effects. And mTORC was subsequently discovered when researchers were probing how rapamycin was achieving these therapeutic outcomes. And when this mTORC protein, which is actually a kinase, is inhibited by rapamycin, it induces autophagy, and it's also known to extend lifespan. And so this is a very active field in molecular biology right now. So mTORC, again, is a serine threonine kinase, and it functions at the crossroads of anabolic and catabolic metabolic programs, meaning mTORC dictates whether a cell is breaking down molecules for food, called autophagy, or building more complex molecules such as DNA or protein for proliferation. mTORC inactivation is necessary and sufficient to induce autophagy. So inactivating mTORC is necessary and sufficient to induce autophagy. And it also significantly downregulates translation. And the wording here is important. The necessary component means mTORC must be off in order to induce autophagy. And the sufficient component of that sentence means inactivating mTORC alone is enough. It's sufficient to induce autophagy. So being necessary and sufficient to induce autophagy is a pretty big deal. Defects in mTORC signaling have been implicated in many different neurodegenerative diseases, such as like Alzheimer's disease or Huntington's disease. Just about every neurodegenerative disease has some line of research into mTORC signaling. In terms of where mTORC signaling occurs, it's actually coordinated from the surface of the lysosomes. And this has basically earned lysosomes as their newfound title as being metabolic signaling hubs. So lysosomes are originally kind of just garbage disposals. They weren't too interesting as organelles. But since the discovery of mTORC and its really complicated regulatory networks, lysosomes have become much more interesting, and they're considered the metabolic signaling hubs of the cell. And lysosomes themselves are actually dynamically shuttled between the plasma membrane during basal high nutrient available conditions. And the perinuclear area is where we see lysosomes during nutrient deprivation. So we see a dynamic shuttling of lysosomes to the plasma membrane during high nutrient availability. Then we see the perinuclear clustering of lysosomes during nutrient deprivation. So this is the general overview of mTORC signaling, and now we're going to talk about some of the specifics in terms of actual pathways. Okay, so we're going to begin our discussion with mTORC. And so mTORC, mammalian target of rapamycin complex, is a complex. It consists of a couple different proteins. I think most importantly is mTOR. mTOR is the basic component of the mTORC complex. We also see really important subdomain or subunits including raptor. Raptor's very highly regulated. Deptor and mammalian LST8. We're not going to go through exactly what each of these domains are, but I just want to give you some overview that mTORC isn't just a single protein, it has multiple domains. And let's look at some of the activities of this serine threonine kinase. In terms of autophagy, autophagy is a major line of research right now, and mTORC is critical to the inactivation of autophagy. So when mTORC is turned off through whatever mechanism, we're going to talk about a few later, when mTORC is off, it relieves this inhibition on this protein called ULK1. And ULK1 is the primary, it's a pre-initiation complex of autophagy. And so by inhibiting ULK1 via phosphorylation, mTORC prevents autophagy initiation. And this is basically the main mechanism by which mTORC prevents autophagy. Another mechanism by which mTORC prevents autophagy is by inhibiting a key transcription factor called TFEB. TFEB is the primary transcription factor that activates pro-autophagic gene expression in the nucleus. So this will be the nucleus. Normally, when TFEB is allowed to enter the nucleus and initiate gene transcription, we see the upregulation of ATG or other autophagy-inducing genes. So mTORC inhibits TFEB. And specifically, mTORC does this by placing a little phosphate, and this recruits this inhibitory 1433 protein that binds and inhibits TFEB. So technically, mTORC phosphorylates, and that recruits this 1433 protein. It's a serine threonine binding protein that will bind to TFEB and basically cover up its nuclear localization signal and prevents TFEB from entering the nucleus. So mTORC is preventing autophagy by inhibiting ULK1 via phosphorylation and causing TFEB to bind to 1433. And that prevents TFEB from entering the nucleus and activating autophagic gene transcription. It also, TFEB also initiates lysosomal biogenesis. It does this through the binding these so-called clear elements, CLEAR elements. Anyways, what about translation? Translation, I said, is upregulated when mTORC is on. How does it do this? Well, let's go with this color. So mTORC actually phosphorylates a protein called 4E-BP for EIF4A binding protein, or I can't remember exactly what the 4E stands for, but this is a protein that inhibits translation. It binds to the 5' cap of mRNA, and it prevents that cap from recruiting EIF4G. And so basically, EIF-BP or 4E-BP inhibits translation. And what this mTORC phosphorylation does, when mTORC phosphorylates 4E-BP, it prevents it from binding to the 5' cap of mRNA, and thus it's promoting translation by inhibiting the inhibitor of translation. It gets a little bit confusing, but 4E-BP is inhibited, and this allows translation to continue. And actually, a lot of viruses do something very similar. Viruses will inhibit 4E-BP, and so viruses do that so they can be translated. If a virus didn't inhibit 4E-BP, the cell would stop translating, and the virus wouldn't be turned into, the envelope proteins wouldn't get made, and the virus wouldn't be able to escape. So 4E-BP is a really important protein when it comes to translation. There's one last critical player here, and this is going to be S6K. So S6K is another kinase that is pretty much always active whenever mTORC is active. So mTORC activates S6K. S6K is the ribosomal S60 kinase, I think, or something like that. It's a kinase that targets the ribosome. It's a kinase that targets the ribosome and activates it. And it leads to translation. So S6K is interesting because it's pretty much always active when mTORC is active, and it's actually the amount of phosphorylated S6K is a proxy for how active mTORC is. So if a researcher wanted to see how much mTORC activity was going on in a cell, he or she would look at phosphorylated S6K, because this is immediately downstream. It's highly correlated to mTORC activation. And S6K achieves increased translation by phosphorylating the ribosome in such a way to activate translation, leading to more translation. Okay, so we've been talking about mTORC and how mTORC inactivates autophagy through ULK1 and inactivating TFEB. And mTORC also activates translation through S6K activation or 4E-BP inactivation. So how does mTORC itself get activated? Well, I think the best place to start when we're discussing mTORC activation is going to be the plasma membrane. There's a couple of different ways to activate mTORC, but I'm going to start with the more conventional mechanism of activating mTORC. And it begins with some kind of growth factor. That's right, GF. It's a growth factor, and it binds some kind of receptor tyrosine kinase. So I'm just going to call it an RTK. So receptor tyrosine kinase binds growth factor. It's a pretty conventional pathway. They autophosphorylate each other. And the phosphorylations that each of these receptor tyrosine kinases put on each other recruits a protein called it recruits a PI3P, or phosphoinositol 3 kinase. It's a kinase that phosphorylates a membrane phospholipid, phosphatidylinositol, at the 3' position. So let's say these are phosphoinositols that have been phosphorylated at the 3' position. And this produces a binding site for certain kinds of proteins, like PDK1 and PDK2. When these proteins are recruited to the plasma membrane because of this PI3Ps, it allows it to these PDK1 and 2s phosphorylate another protein called AKT. So AKT gets phosphorylated and activated by PDK1 and PDK2. So this causes the activation of our AKT, which is a really important growth signaling kinase that is downstream of RTKs. And when AKT is activated, it inhibits a protein via phosphorylation called TSC1/2. And TSC1/2 is a Rheb gap, meaning it hydrolyzes the GTP of Rheb into GDP. Oops. Let me fix that. So it's inactivating Rheb by hydrolyzing its GTP into GDP. So this is kind of a double negative. AKT is inhibiting this other protein, TSC1/2. TSC1/2 then inhibits Rheb. So an easier way to think of it is AKT activates Rheb. Although it doesn't do this directly, it does it through TSC1/2 inhibition. And AKT is again a kinase that's doing this via phosphorylation. And Rheb is actually a lysosomal bound protein. So this is going to be our lysosome. This activation of Rheb occurs on the lysosome. And when Rheb has GTP, when it has GTP because TSC has been inhibited, TSC likes to turn Rheb-GTP into Rheb-GDP, which is the inactive form. But if TSC is gone, then Rheb is going to have GTP. And Rheb, when it's bound to GTP, can recruit some effector proteins. One of the effector proteins for Rheb-GTP is going to be mTORC, our critical protein. So mTORC is being recruited to the lysosome by Rheb. So Rheb, now when it's bound to GTP, is the reason mTORC is found at the lysosome. And so through these different steps, we see AKT signaling leading to the activation of mTORC and the inhibition of autophagy, activation of translation. Just a really quick correction. Rheb activates mTORC through an unknown mechanism. I said that Rheb recruits mTORC. Well, it doesn't actually recruit mTORC, but it is required for mTORC activation, and the mechanism is not known. So I just wanted to add that the Rheb-GTP doesn't necessarily recruit mTORC, but Rheb-GTP is required for mTORC activation at the lysosome. Okay, so we talked about how AKT can lead to the activation of mTORC through Rheb, but we haven't actually discussed how other things can activate mTORC, like amino acids. Amino acids are critical to the activation of mTORC. So let's begin with our amino acids. Let's go through the pathway of how amino acids activate mTORC. So let's say our amino acids enter the cell via macropinocytosis, very common. Pinocytosis means water drinking in Latin, so it's literally the cell drinking the extracellular fluid. And these become vesicles, and the vesicles get delivered commonly to the lysosome. This will be our lysosome. And so when these amino acids get delivered to the lysosome, you now have lots of amino acids in here. You could also have amino acids perhaps from degrading cellular components. But whenever there's lots of amino acids around, I don't know if the mechanism has been determined, but they activate this protein called a VATPase. VATPase. And it may be that the exit of amino acids somehow activates the ATPase because it is a channel protein. And nevertheless, when VATPase gets activated, it activates Ragulator. Ragulator sounds like regulator, and that's because it regulates mTORC activity, as we're going to see in a second. So the Ragulator is kind of a confusing protein. The Ragulator functions to control two different proteins. Regulator is a GTPase that essentially controls the fate of these two very similar proteins called RagAB and RagCD. And RagAB and RagCD can both be bound to ATP or ADP, depends on the circumstance. But when Ragulator is activated by this VATPase, it gives GTP to RagAB, and it gives GDP to RagCD. When RagCD has GDP, it's inactive. And when RagAB has GTP, it's active. So this is going to be an active protein now. And the reason this is important is because RagAB actually recruits mTORC. So I mentioned this in the last slide. I said something like Rheb recruits mTORC. I meant RagAB, when it's bound to GTP, is what recruits mTORC. It's the effector for mTORC. And Rheb is simply there to activate mTORC once it arrives. So it's kind of a two-step activation process. mTORC needs to be recruited to the lysosome by Ragulator's activation, or I should say guanine exchange factor activity of RagA. And it also needs to be activated by the presence of GTP Rheb. So say this is GTP Rheb. So I should also mention that this RagCD complex, if it's not being inhibited by Ragulator, inhibits mTORC. It inhibits mTORC's translocation to the lysosome. So let me reemphasize everything. This is kind of a confusing pathway. We have amino acids that come in through here. The amino acids can potentially exit, and when they exit, they turn this turbine, this VATPase, and this somehow activates Ragulator. When Ragulator is activated through this mechanism, it gives RagA GTP, RagA-GTP, and it simultaneously inactivates RagCD. When RagA is bound to GTP, it can recruit mTORC to the membrane of the lysosome. Right here, we see mTORC has been recruited. It's recruited by RagA. When RagA recruits mTORC, mTORC actually comes in an inactive form. And in order to be completely activated, it needs to bind to GTP Rheb. So there's kind of a two-step activation. We have not only does mTORC need to be recruited by RagA, but GTP Rheb needs to be activated as well and ready to accept mTORC and activate it once it arrives. And once both these conditions are met, then we get autophagy inhibition, we get translation. So if you have any questions about this pathway, be sure to comment. So in this slide, we're going to be talking about something slightly different. We're going to talk about the localization of lysosomes as it relates to mTORC activation. So under basal conditions, under just normal business as usual conditions, we have all these are just different receptors embedded into the plasma membrane of a cell. And these receptors are actually continuously degraded or turned over by lysosomes. Lysosomes near the plasma membrane are just kind of continuously degrading receptors. And in addition to this, we have our mTORC. And mTORC is typically active when it's bound to peripheral lysosomes. And we'll discuss why that is in a second. But for now, this is kind of the general what's generally happening when nutrients are available. Lysosomes sit near the plasma membrane. They continuously degrade receptors. They hang on to active mTORC. I'll say mTORC on. And then something happens during starvation. So let's say this is a microtubule. And the various, we're going to talk about retrograde transport. So starvation, these are things that can actually cause the retrograde transport of a lysosome. Typically, it's just thought of as starvation inducing the retrograde transport of lysosomes. But interestingly, you also have mutant Huntington and LRKK2, which is a protein mutated in some Parkinson's disease patients. But basically what's happening is that starvation causes the retrograde transport of lysosomes back to the periphery, or sorry, the cell nucleus, the Sumir nucleus. So the question is, how are lysosomes actually transported retrogradely towards the nucleus? And that's what I want to cover in this slide. Well, lysosomes are attached to this protein called Rab7. Rab7 is a protein that associates with lysosomes because of the type of phospholipids that lysosomes have. Lysosomes are marked by PI3P, I think. phosphatidylinositol phosphorylated at the 3' position. And that's what recruits Rab7 to these lysosomes, is that they're recruited in that way. And in order to be transported retrogradely, lysosomes need to some way link up with dynein and dynactin. Dynactin is associated with dynein and serves as kind of an adapter. So in order to be transported retrogradely, this Rab7 lysosome needs to somehow link up with dynein, dynactin, to be moved back to the nucleus. So how does that happen? It occurs through an adapter protein. So we're talking about a lot of adapters here. But dynactin, which is linked to dynein, can link to Rab7 through this other protein. This is going to be our adapter protein. It's called RILP, Rab7 interacting lysosomal protein. So it's what it is it's just an adapter that links Rab7 to dynactin. Dynactin is linked to dynein, and through this mechanism, we get retrograde transport. I guess it's kind of confusing as to what actually activates the retrograde transport. I said starvation, but how does starvation suddenly cause this retrograde transport to occur? And it's thought it might have something to do with lysosomal pH. A loss of lysosomal pH, when lysosomal pH increases, it can cause the retrograde transport. And the lysosomal pH is a function of its internal amino acid content, because amino acids can actually fuel, or I should say that it can power a VATPase. It can power a proton pump, which affects lots of things like ion concentration and its membrane potential and obviously pH. But I'm not too sure about that, so I'm not going to get into it. Let's talk about what happens once lysosomes, let's not use green, use blue. So let's say the lysosome arrives down here at the nucleus. This happens during starvation. And when our lysosomes are perinuclear around the nucleus, mTORC is either dissociated or it's off. Actually, it's usually dissociated, but I'll just write it here as if it was on or attached to the lysosome. But mTORC is off when it's located around the nucleus. And this has important consequences for things like autophagy. If we have an autophagosome, which is now going to be activated because mTORC is off, this allows our lysosomes to fuse with our perinuclear autophagosomes. And so this is actually typically the mechanism by which lysosomes are brought in close proximity with autophagosomes, is because they're both located around the nucleus, so it allows them to interact. If the retrograde transport of these lysosomes doesn't happen, they typically can't find their autophagosomes, and they don't fuse, and autophagosomes are stuck around the nucleus, not being able to degrade their contents because they can't find the lysosome. So another consequence of mTORC turning off is it detaches TFEB. So TFEB is allowed to enter the nucleus. We talked about the transcription factor that binds clear elements, and it's normally constitutively inhibited by mTORC. When TFEB, or when mTORC is off, it allows TFEB to enter the nucleus and prime gene expression. And one of the a very recent paper found a specific mRNA and protein that is upregulated following TFEB translocation to the nucleus. And this protein, TMEM55 for transmembrane 55B, so it doesn't really have a name yet, so it's that new. What they found is this TFEB induced protein, TMEM55, and somehow promotes the dynein retrograde movement of lysosomes. So when mTORC is off, it promotes the transcription of TMEM55B, which can promote retrograde transport. They don't in the paper they didn't specify how that might be happening, but TMEM55 does increase the rate of retrograde transport. So this is probably a mechanism by which a couple of lysosomes being transported retrogradely can trigger the appropriate gene expression to induce retrograde transport of all the lysosomes. So this is a pathway by which lysosomes are transported back towards the nucleus so they can fuse with autophagosomes. Now let's discuss how the lysosomes are moved back to the cell periphery when nutrients are available. So this is going to be another microtubule. And in order for a lysosome to be moved back towards the cell periphery, where it's going to be active, or mTORC is then going to be reactivated. We'll see how that happens. There are a couple complexes involved. The BORC complex, BORC complex, and the RL8, the RL8 GTPase. So they form a complex on perinuclear lysosomes. And so BORC somehow is recruited first to lysosomes. BORC then recruits RL8. And RL8, when it's bound to GTP, binds to an effector. The effector for RL8 is this other protein called SKIP. Let's see if I can... SKIP, S-K-I-P. And SKIP is another adapter, kind of like RILP, but SKIP links up to kinesin. Kinesin is our plus in motor protein on microtubules, kinesin 1. And so we have this BORC bound to, we have BORC bound to lysosomes. It recruits RL8, which then somehow gains ATP. It might be a member of the BORC complex that functions as a GEF, I'm not sure. But RL8, when it's bound to GTP, interacts with an effector called SKIP, and SKIP links the complex to kinesin. And this allows the anterograde movement of lysosomes. And once our lysosomes get to the periphery of the cell, this will be our lysosome now, it allows mTORC to be active, or mTORC eventually ends up getting activated because where it's located. Specifically, we have our receptors over here that are going to be activated by growth factors. Let's just say like insulin. Insulin is a classic growth factor, right? And insulin triggers signaling through AKT, which is, we talked about earlier, protein kinase B is another name. And AKT activates mTORC. So what's important is that AKT is typically most active in the periphery of the cell because that's where the receptors are located. And AKT is activated right around here, right below the right next to the membrane. So AKT doesn't diffuse very far. So mTORC and AKT need to be in close proximity in order to interact. So this is a general pathway for how lysosomes are moved towards the nucleus and then back out to the periphery. Let me just reiterate. So we have lysosomes over here in the cell periphery. Loss of amino acids, starvation somehow causes the retrograde transport, and it functions through Rab7. Rab7 binds to RILP, which interacts with dynein, a component of, dynactin, which is a component of the dynein complex. And this triggers the retrograde transport of lysosomes. When lysosomes are at the nucleus, mTORC is typically turned off because it's been dragged away from its growth factor signaling. And this releases TFEB. TFEB can enter the nucleus and upregulate things like TMEM55. TMEM55 promotes more retrograde movement of lysosomes. The mechanism isn't known, but it is an important factor. In order for lysosomes to be moved back out to the periphery of the cell, it requires this complex called BORC. BORC interacts with RL8 and perhaps functions as a RL8 GEF to give it GTP. GTP bound RL8 can bind to skip. Skip is an adapter for kinesin 1. Once it's linked up to kinesin 1, it can be moved back out to the periphery of the cell. And when the lysosome is moved back out to the periphery, right here, it can turn, it leads to the activation of mTORC because it's now co-localized with active AKT. Okay, so let's review this excellent paper by Hong et al. Titled Phosphatidylinositol 3-phosphate controls mTORC signaling through lysosomal positioning. So previous research has found that the PI3K, called VPS34, can be activated by amino acids, and this causes the activation of mTORC. So VPS34 is a major phosphatidylinositol 3 kinase that is also critical for autophagy, but we won't get into that. In this case, VPS34 is somehow activated by amino acids, and this leads to the activation of mTORC, which makes sense because when amino acids are present, mTORC should be on and promoting translation. But the specific mechanism by which VPS34 is activated by amino acids and how VPS34 activation leads to mTORC activation is not understood. Perinuclear lysosomes, which are observed during states of starvation, are associated with decreased mTORC activity, decreased lysosomal fusion, and decreased autophagy, while peripheral mTORC positive lysosomes are known to promote mTORC activation. Now, these two ideas, VPS34 activation and lysosomal positioning, were in no way related until this paper came out. And that's why this paper is so cool, is that it ties together these two different events and concepts and tells a more coherent story of how mTORC signaling actually works. So this is what they discovered. First, a lysosomal bound kinesin adapter called FICO1 is recruited to perinuclear lysosomes following the activation of VPS34 bioamino acids. So let's think about that sentence again. So VPS34 is activated by amino acids, and this recruits FICO1 to lysosomes. And FICO1 functions as an adapter to kinesin, essentially attaching lysosomes to kinesin, the kinesin motor protein, which you probably remember moves cargo to the plus end of microtubules, meaning this event would move lysosomes towards the periphery of the cell. Once VPS34 phosphorylates the lysosomal membrane phosphatidylinositol and recruits FICO1, FICO1 then interacts with another protein called protrudin. Protrudin is a resident ER protein. Remember, the ER is perinuclear, so these ER lysosomal contact sites would only be occurring during starvation when lysosomes are also perinuclear. This FICO1 protrudin interaction is important because protrudin is responsible for handing off kinesin to FICO1. So even though FICO1 is a kinesin adapter for lysosomal transport, it doesn't grab kinesin by itself. It actually needs protrudin to kind of facilitate that interaction. And ultimately, this protrudin FICO1 interaction is required for peripheral localization of lysosomes and full mTORC activation during nutrient deprivation. Because when FICO1 or protrudin is inhibited or knocked out or whatever, lysosomes fail to move to the periphery of the cell, and mTORC is deprived of growth factor signaling and is not nearly as active as it should be. So This is the summary of the current paper. We're going to go through the pathway that is being put forward in the next slide. Okay, so this pathway, let's begin with the entrance of amino acids. So let's say some amino acids are present and they are entering the cell. The amino acids come through, they enter the cell, and they activate our VPS34, our phosphatidylinositol kinase. And I also want to add that this VPS34 that's being activated is in complex with UVRAG, which is important to note because VPS34 is also critical for autophagy. And this pathway that we're talking about is basically inhibiting autophagy. So the authors had to deal with this conundrum where VPS34 was both critical for autophagy and mTORC activation, which are completely diametric processes. But anyways, the problem is probably due to UVRAG. The specific proteins are in complex with VPS34. Anyways, let's not get caught up on that. So VPS34 is activated by amino acids. VPS34 turns to lysosomes. So let's say this is our lysosome. And what VPS34 does is that it begins phosphorylating all the phosphatidylinositols at the 3' position. So all these little marks I'm putting, these are basically phosphatidylinositols that have been phosphorylated at the 3' position. That's what I'm indicating by these. So lysosomes get phosphorylated at the 3' position. And this is occurring at the perinuclear region. So this is our nucleus. So these are inactive lysosomes next to the nucleus. We talked about how that's, there's inactivated the mTORC. mTORC is off at this point, right? Because the lysosome was next to the nucleus. That's correlates with inactive mTORC signaling, which means that we have TFEB going into the nucleus. I just wanted to mention that, remind you guys. So TFEB is entering the nucleus, priming gene expression. Now, this is where the pathway picks up, is that they're looking at this, this protein called protrudin. Protrudin is so this is our endoplasmic reticulum, our ER. And protrudin is a protein that protrudes from the endoplasmic reticulum. It is bound to the ER. It is an integral membrane protein of the endoplasmic reticulum, protrudin. And protrudin actually interacts with lysosomes because, let's not use green, because of these phosphorylated phosphatidylinositols. So protrudin can bind to lysosomes using a 5e domain, FYVE. These 5e domains bind to phosphorylated inositols at the 3' position. And so this allows protrudin to interact. So protrudin, which is inside of a membrane protein of the ER, to interact with lysosomes when they are near the nucleus. This facilitates the ER lysosomal contact sites. The phosphorylated lysosomes also recruit our FICO1 proteins. FICO1 is another protein that contains a 5e domain, and it interacts with lysosomes. So FICO1 binds lysosomes through the 5e domain. And this interaction puts protrudin and FICO1 in very close proximity. And there's also an interaction between protrudin and FICO1. And so these 5e domains and this interaction between protrudin recruits FICO1 to lysosomes. And the reason that's important is because FICO1 is a known adapter for kinesin 1. So these lysosomes are being connected or attached onto chysin through FICO1, and this allows the antegrade transport through via microtubules. So this protrudin is functioning to recruit FICO1 to these ER lysosomal contact sites, and the FICO1 functions as an adapter to chysin 1. And this entire process is due to VPS34. VPS34 is what places those membrane lipids that interact with the 5E domains and allows the antegrade movement. So really the amino acids are activating VPS34, which is what is responsible for this movement of the lysosomes back towards the cell periphery. Remember, the cell periphery is where AKT signaling is. So when the lysosomes get out here, so this is a lysosome, and it's going to be pulling our mTORC right next to this AKT signaling, and our mTORC is going to be on. And this is how mTORC gets turned on. And this is the general pathway that's being put forth by the current study. PI3K inhibition prevents mTORC activation and FICO1 localization to lysosomes. So in this left figure over here, we see that the typical peripheral localization of mTORC right through here, this is what mTORC typically looks like. It's more peripherally located. It's actually disrupted when SAR 405 is administered, and SAR 405 is a VPS34 inhibitor. So when the VPS34 phosphatidylinositol 3 kinase is inhibited, it prevents the peripheral localization of mTORC. We see a similar idea over here on the right, is that VPS34 inhibition prevents FICO1 from interacting with lysosomes. There isn't any co-localization between FICO1 and lysosomes. So in other words, VPS34 is required for FICO1 to localize to the lysosomes and for mTORC to be transported out into the periphery of the cell. Protrudin promotes peripheral mTORC localization. So protrudin was shown to bind FICO1 and to lysosomes through a 5E domain. I don't have that shown here. And transfecting protrudin, as seen right here, just a very simple transfection of protrudin, significantly decreased the intensity of perinuclear mTORC. So protrudin, when it's upregulated, decreases perinuclear mTORC and promotes the movement of mTORC to the periphery of the cell. So you can see that, let's see, MYC, so this is protrudin transfection. You can see lots of mTORC. If you zoom in right here, you see lots of mTORC around these edges right here. This is very active mTORC signaling. And when protrudin has a mutated 5E domain right here, when the 5E domain has been mutated, you do not see mTORC localizing to the membrane. You just see dispersed mTORC signaling. And also this domain is KIFBD. This is the domain that protrudin interacts with FICO1 through. So when protrudin cannot interact with FICO1, it also results in dispersed mTORC signaling. So in order for protrudin to facilitate this this nice peripheral localization of mTORC, it requires a 5E domain to interact with the phosphorylated phosphatidylinositols, and it requires this KIFBD domain to interact with FICO1. See, protrudin activates mTORC signaling by co-localizing mTORC positive lysosomes with peripheral phosphorylated activated AKT. So we discussed earlier, I've been talking about how AKT activates mTORC, and the problem with that is that it comes down to physical location, because AKT is activated on the membrane, while mTORC can be dynamically shuttled between the perinuclear region and the peripheral membrane area. We see that when in this slide to the left over here, we see, again, this is proof that what I've been talking about is real, is that AKT, when its phosphorylated active form is over here in the periphery, you can see lots more phosphorylated AKT over here in the periphery. And we also see some lysosomes in red. You see these little lysosomes that are co-localized, and any mTORC on these lysosomes are getting activated. This is in contrast to what's happening over here on the right. This is protrudin has been knocked down using siRNA. So when protrudin is gone, you don't see any lysosomes in red. In red, it's supposed to be lysosomes. I don't see any lysosomes over there. All the lysosomes appear to be around the nucleus. So there's no mTORC getting activated in these cells because the lysosomes aren't moving to the periphery. So I hope you guys enjoyed this paper and this lecture. If you have any questions at all or notice an error, please let me know. And thanks for watching.